Professor, I have a quick question.
Oh, yeah, sure.
So I was reading the syllabus and it says that midterm 2 only covers lectures 7 through 12. So will lectures 13 through 15 that we're doing today not be on the midterm?
Yeah, you're right. So yeah, today's lecture will not be covered for the second midterm. So that's why. So today I'm gonna spend like half of our time to cover to teach the new stuff. But this will not be on the second midterm, and we're going to use the second half of the time to go over the practice test. So yesterday I uploaded a practice test for the second midterm to the iLearn. I hope everyone have a chance to review it. Okay, yeah, good question. Thank you. Yeah, today's stuff will not be on the second midterm, but definitely it will be on the final. Yeah. All right. Let's get started. Let me see. Okay. Okay, so last time we started talking about the abnormality in a larger scale compared to the previous stuff, right? The previous mutation is very occurs in a very small region, like a point mutation, just a change in a single nucleotide, or a small region, like the duplication of a small piece of DNA or gene. But now from lectures 13 to 15, so we are focusing on the large scale mutation, which involves like either the entire chromosome or entire sets of chromosomes. Okay. All right. So let's continue. So here, so when we talk about a trisomic, so trisomic means like 2n+1, right? For example, in a diploid organism, so trisomic means like there's an extra copy of one specific chromosome. So if this is the case, so for example, in this organism, trisomic organism, if there are 3 copies of the same chromosome, so what would happen during the meiosis? So during the meiosis, these 3 homologous chromosomes are going to be pairing together like this. There are 2 possible ways for them to pair. Either 2 of them will be paired in a normal way and leaving the other one alone. And then, so this 2 is gonna be separated, right? And the spindle fiber will pull these 2 chromosomes, homologous chromosomes apart, going to 2 different daughter cells. And this one will randomly going one way, for example, going to this pole, and then so the product from this meiosis will end up with 2 daughter cells, right? One has just this copy. All right, so we have a chromosome here, but the other one will have 2 copies, right? This is another gamete. Okay. All right. So this is one possible way for the pairing. Another possible option for these 3 homologous chromosomes to be paired together. So it's like this. We call this form as the trivalent, like this. And so during the meiosis, this one, look at this one. So this one is going to this direction. The spindle fiber will pull this one towards this side, and this two towards the other direction. So end up with a gamete with one copy here, and another gamete with two copies. So this is how the trisomic. So this is how the chromosome will be. The three copies of the homologous chromosomes are going to segregate in a trisomic individual, right? So I hope this is clear. So so let's look into the detail. All right, so this is a little bit advanced compared to what we have learned before, right? If in a regular diploid organism, so if we have like one chromosome, the other chromosome, and on this chromosome there's a gene. So suppose there's a dominant allele and there's a recessive allele. So this parent will produce two possible gametes, right? One is the big A, and the other one is the small a with equal chance. So we have learned this before, and we can use the Punnett square to help us to dissect the genetics behind the monohybrid cross, right? This is for one gene. However, in the trisomy case, we have three chromosomes, homologous chromosomes, and suppose one chromosome has a dominant allele on at this loci, and the other two has the recessive. So so if a parent is like this, the trisomy, so what kind of like gametes can this parent produce? Okay, so first, we have learned this before, right? In the previous slides, there are two possible ways to do this. To do this, let's use this way. They are equivalent. Let's consider this option here, and look at here. So first, if so, we label these three chromosomes as 1, 2, 3. So if 1 and 2 come together into a daughter cell, that means simultaneously we have a daughter cell like this, right? The gamete like this, all right? So if 1 and 3 are going together and end up being in the same gamete, then simultaneously we're going to have a gamete with this, right? And so on. So those are the all possible outcomes. And if you summarize this, all these outcomes, you're going to see like from this parent, the trisomy parent, this parent can produce 4 types of gametes with a single capital A, gametes with single lowercase a, and heterozygous, and a gamete with little a, and the probability that the probabilities that are associated with each of these gametes will be this. So this is not difficult to understand, right? So if you can figure out this, and then so it's easy to get this information. So, all right, for a trisomy parent with the genotype like this, the big A, small a, and this will be the possible gametes that this parent can produce with the probability like this. Okay, so this is for the trisomic case, and for the polyploidy case, so that's the change in the number of the entire sets of chromosomes, right? We have learned this before. The monoploid, or also we can say this is haploid, single copy. So that's the one set of chromosomes, the entire sets. Okay, haploid. Okay, and the diploid, triploid, and so on. Right. Okay, so the other day, last time, a student asked me a question. So why polyploidy can be tolerated in plants, but not in mammals, in animals? Yes, yeah, that's the reality. That's the truth. But there are many like explanations behind this. So I sent out. I did some search, literature search online, and I found some interesting and useful explanations. And I send a announcement to everyone. So hopefully you have time. If you have time, you can read it and read them through, and this can help you get some idea about this. Indeed, so the polyploidy are not tolerated in human. So only 2N diploid are viable in humans, right? And also polyploidy is very rare in other animals. For some lower level of the animals, probably polyploidy may be tolerated. For example, in frogs and reptiles, they sometimes they can survive if they have like more than 2 copies of their chromosome sets. And but this is very common in plants. So you can see probably like more than 50% of the situation. So more than 50% of the plants, they can tolerate more than 2 copies of the chromosome sets, 2 sets of the chromosomes. Right. All right, for example, banana is actually the 3 copies, 3N and triploid. Right. Okay, so in plants, what's the benefit? What is the benefit to have more than 2 sets of chromosomes? Okay, the higher ploidy will produce larger size. If we, so this is example. So if we look at the diploid and the tetraploid grape, you're gonna see, yeah, if we put them side by side, you're gonna see a difference, right, immediately. So this is much larger than this. This is because of the different dosage of the gene expression. Because let's think about in this way. So for a diploid on a genome for a specific gene, so there are only 2 copies of that gene, right? But in the tetraploid, they have 4 copies. If all these 4 copies of the genes can be expressed, that means like they can produce like doubled gene product in the tetraploid. So no wonder they can grow faster and larger. Yeah, that's the benefit to be to have higher ploidy in plants. The same thing here. So this is the stoma, 2N. The diploid is smaller, 4N is larger, and 8N. Yeah, you can see the difference, right? So this table just gives us a summary about what we need to know so far in this lecture, in lecture 13. So we need to understand the difference between the euploids and the aneuploids. Euploids means like we have the exact multiple of the haploid number of the chromosomes. So either the monoploid, the haploid, 1 copy of the genome, so which is 1N, diploid 2N. So if we have 3 chromosomes in the haploid A, B, C, Then diploid, we're going to see doubled, right? The two chromosome A and two chromosome B and two chromosome C. And this is the total number of the chromosomes. Triploid, 3N, then you're going to see 3 copies of each chromosomes, and the total number should be 9, and so on. So still they are balanced because they are exact multiple of the haploid number of the chromosomes. But if we look at the aneuploids, so that's different. So usually like this involves like 1 chromosome missing, which is 2n minus 1, or 1 chromosome extra. So this is the trisomic 2n plus 1. So this is like a monosomic chromosome A, right? And this is the trisomic B here. Okay. So as I mentioned, so the difference between different ploidy or different or between the monosomic and trisomic. So that's so if you see different phenotypes due to this different genetics, that's mainly because of the gene dosage effect. Because if you have different copies of a specific gene, then you're going to see the different phenotypes, right? If that phenotype is associated with a specific genes on that chromosome. Okay, so this gives us a summary. I try to link everything we have learned so far together. Before the midterm one, we learned just one gene inheritance pattern. So this is a standard way, right? We talk about the diploid organism. So if we have a monohybrid parent and another parent, so we can easily use this Punnett square to help us to understand the genetics behind this cross. And this will give us a classical 3-to-1 ratio if we have the dominance and the recessive relationship between these two alleles. All right, then we expand this a little bit to talk about 2 independent genes. So now we have 2 genes. They are independent. They are on different chromosomes, unlinked. So if this is the case, so when we do the dihybrid cross, we still can use the Punnett square, and this gives us the classical ratio of 9 to 3 to 3 to 1, right? So this is what we have learned before midterm 1 and midterm 2. So we try to expand this more. So we studied the relationship between 2 genes. So if the 2 genes there's no interaction between these 2 genes, you're going to see this pattern as well, just like this. They are independent. However, if the 2 genes are somehow related to each other by some pathway, biological pathway, so depending on their relationship, so we're going to see different ratios if we do the dihybrid cross, right? So this ratio will indicate that these 2 genes in the same pathway, and or so this ratio will indicate the recessive epistasis and so on. So okay, right.
Professor?
Yep.
Would it be possible for you to provide an example when 2 genes are in the same biological pathway?
Oh, you mean this? This is a duplication, right? So yes. Let me go back.
Thank you.
Let's see, the same pathway. So let's go back to, give me a second. Let me stop share here and try to find out. Here. I hope this is the correct slide. No, this is not the right one. Give me a second. Sorry. Probably this one. So yeah, this is the, so can you see my slides here? All right, so the complementation, complementary gene actions of the two genes will produce this ratio, 9:7 ratio in the dihybrid cross. So for example, like this one. Okay, so yeah, this is a perfect one. So we're talking about like two mutant flower plants. This one, so the wild type is like the wild type means like oops. Okay, so the wild type means like so these two genes, the gene 1 will produce the functional enzymes A, and the gene 2 will produce functional enzyme B. So if in a wild type individual which can produce both functional A and B, then it can transform the colorless X into colorless Y, and finally you're going to see the purple end product. So this will make us to see purple color in the flower petal. However, so this means like these two genes are in the same pathway, right? All right. However, if like one gene has been mutated, or the other one has been mutated, then you're going to see the mutant type, right? You're going to see only the white color flower, or if both genes are mutated, right? So, and let's look at here. So if we cross those two, this is the dihybrid cross, right? Oh no, sorry. If we have two parents, the mutant parents, when we cross those two, we're gonna get the F1 dihybrid individual. If we intercross this one with another plant of the same genotype, then we're gonna get this, right? Because this is the wild type, but all this will give you the mutant phenotype. So if you look at the ratio of this will give you the 9:7 ratio. So is that clear?
Yeah, that's clear.
Okay, right. So this is very important, and definitely this part will be on your next midterm, which is on Tuesday. So I hope like so during the weekend, please spend some time on this to make sure you understand the biology, the mechanism behind each different ratio. So we start from the very basic ratio, 9:3 to 3:1 ratio. That's the ratio shows no interaction between these two genes, right? And we're going to see we're going to see the variant of this ratio because of different relationship between these two genes. So this is one example. If these two genes are involved in the same pathway like this will give you the 9:7 ratio. Okay. Yes, very important. So this is a very important stuff. Okay. So yeah, so okay, so midterm 2. This is midterm 2. So we're gonna talk about this, right? We're going to focus on this. So this is one of the important topic we're gonna test on the second midterm. Of course, we're going to test other things like the genetic mapping. So how so if we do the 3-point test cross, and we got a bunch of like data. So how do you calculate the genetic map distance? How do you derive the map for the 3 genes, right, on the same chromosome? And how do you calculate the genetic distance between each pair of the genes? So that's another topic. And also we use the neurospora, the haploid organism, model organism, to do the mapping analysis. That's another major topic for the coming midterm. Okay. All right. So this is for the final exam. So today's stuff, but yeah, it is not so critical for now for next Tuesday, but definitely. So this is important for the final exam. All right. So this is the extension from the 1 gene inheritance in a standard case to a non-standard case. So here we talk about the trisomy, right? So if we have a parent carrying the genotype like this is the trisomy parent, and this is a normal parent. Okay, so we still can use something like the Punnett square. This is not square, this is a rectangle, but it's very similar to the Punnett square, right? We can use a very similar strategy to help us to understand the cross between these 2 parents. We have learned, so for this parent, it can produce 4 types of gametes with this ratios. So yeah, I hope everybody understand this. If not, so after class you can go back to the previous slides to see, to make sure you understand why this is like this. So why this trisomy parent will produce these 4 types of gametes with this ratio. And for the standard parent, it's easy, right? This one can produce half of the gametes carrying this and half of the gametes carrying this. And then they independently, they're gonna produce zygote like this. And then, all right. So if we're talking about 2 alleles, A is dominant, and the big A, the capital A is dominant, and the lowercase a is the recessive. Then if complete dominance, then if we summarize this table, we're going to see the phenotypic ratio should be 9:3, right? Or 3:1, something like this. So, all right. And also we're gonna so in the 3rd part of this course, we're gonna talk about the mutation due to the extra sets of chromosomes. So haploid, diploid, and so on. Okay. All right. So this is example. This could be a question, regular question that I can put on the final exam. Again, so for the final, all the questions will be the multiple choices, because that would be easier for us to grade the test and get the point, get the grade back to you sooner. But for the midterm, right now, using the online platform, so it's not very easy. So, but still, we're gonna take the short answer forms, but I will make it easier for you to type in your answers to each question through iLearn platform. So the question will not necessarily be easy, but the way you answer those questions, I will make it easier for you to type in your answers to each question. All right. So Yeah, and the style for the midterm 2 will be similar to the to that uh, to that for the midterm 1. So you have done a great job in the midterm 1. So yeah, we're gonna take the same form for the midterm 2. For final, so I could give you a question like this. So okay, so there's so either the triploid or trisomic. Let's say trisomic. Trisomic, like only this chromosome carrying this gene will be 3 copies, not the other chromosomes, right? But triploid means like every chromosome will be tripled. So either way. So for example, if we have a genotype for this trisomic or triploid individual like this, and so what is the probability if we do the intercross between these two parents of the same genotype? What's the probability you're gonna see a progeny carrying the same genotype? All right, so that's the question, which means we have 2 parents, and these 2 parents are both, the genotype of these parents are both like this. So if we cross these 2, so what is the probability? So we're gonna calculate the probability that the progeny from this cross will be also like this, the genotype. Okay, so this is a very fair question that I could ask in the final exam. So we have learned this, right? So pairing like this will give you the gametes like this, and if pairing like this will give you the gametes like this and this. So the gamete frequency, so we have learned this before. So each of these parent will produce a gamete like this with this ratio, this, and this. And we have 2 parents here, right? So the 2 parents to produce a progeny of this genotype. So either the first parent will contribute a lowercase a gamete, but the other one will contribute a gamete with the big A, right? Or there's another possibility option. So if the first parent will contribute a gamete with a capital A, but the other parents need to contribute a gamete with this, then you're going to end up with this genotype, right? So we need to use this formula to calculate the probability. So that's the final answer. All right, so this will be a very perfect example for the final exam. Okay. Okay, we still have time. Okay, so if we have a diploid individual, so how can we produce the polyploidy, like the tetraploid individual from a diploid individual? So this is what we can do. Okay, so if we have a regular diploid individual, so for each one you have 2 copies, right? 1 copy and 2 copy. All right. In the mitosis, so during the mitosis, if it is a normal mitosis, so these 2 sister chromatids are going to segregate and end up with 2 daughter cells, each with 4 chromosomes, right? 2N. So each daughter cell will have 2N. So this is the standard normal mitosis. However, if we use something like the colchicine, and this is a chemical that can disrupt the spindle fiber formation. So because to complete a regular mitosis, the cell need to form the spindle fiber, and the spindle fiber is gonna attach to the centromere for each chromatids, right? And pull these two sisters chromatids apart towards two directions to complete the standard mitosis. However, if we apply this colchicine into to disrupt the formation of the spindle fiber, then this sister chromatids cannot separate. They will stay in the same cell. Then what's the consequence? The result is like the cell will have doubled number of the chromosomes. So in this way, we can produce a tetraploid from a diploid like this. Right. Okay, so this is the way how we produce the triploid. So we can easily we can easily produce, based on the previous slides, we can easily produce the tetraploid from the diploid, right, using the colchicine. And then once we have the tetraploid, we can cross this tetraploid with the regular diploid. Now we can get triploid. The same thing here, right? So this is how human can produce different organism with different polyploidy in a lab. Okay, so for example, so I like to eat watermelon. I don't know if you like it. It's delicious, and I think the watermelon season is coming pretty soon. Yeah, it's almost summer now. All right, because when I was a kid, I rarely ate watermelon without seeds. Probably at that time, yeah, we didn't have that technology technique. But now it's easy, right? So usually, so we have watermelon like this seedless. All right, this is because so we can produce a tetraploid watermelon from the diploid, and then we can cross the tetraploid with the diploid to generate the triploid individual. Then this triploid individual, if we cross the triploid individual, they will be seedless. Why? Because if we cross a 3N with another 3N, the only possibility to produce seed is like this parent will produce a 1N gamete, and this parent will also produce a 1N gamete, and then when these two 1N gamete haploids come together, this will produce the seed in the watermelon. Okay. However, because this is a 3N, which means like, so for example, I only use like 4 chromosomes here. So definitely, yeah, in a watermelon, there are more than 4 chromosomes. So I just use 4 as an example. Okay, so in a 3N parent, so each chromosome has 3 copies, right? And during the meiosis, so this one is gonna produce a gamete. So this one, so when this cell goes through the meiosis, so each chromosome will segregate independently, right? So for example, these 3 will segregate independently in terms of this one. So okay, so probably this is one possible gamete, and only 1 copies of the green chromosome come to this gamete. But look at the blue one. So the blue chromosome, unfortunately, 2 copies of the blue chromosome come to the same gamete, and the same thing for here, and this one copy for here. So this is not a 1N. This is a mixture, right? This is not the 1N gamete. And for majority of the gametes produced by these 3N parents will look like this. So they are, those gametes cannot produce any seeds in the watermelon, only if this parent produce a gamete like this. So for each chromosome, only one copy of that chromosome comes together to form this 1N gamete. Then this one has the probability to form a seed. So this is for one parent, and the chance to get a gamete like this, it's only like this. So suppose we have like 10 chromosomes. So the probability to produce a gamete like this is very small. So now this is only for one parent, right? So if we consider another parent, so the chance will be even smaller. So that's why, let's go back to here. So if we want to produce a watermelon with seeds, then the probability should be this. So this is for the 1N gamete for one parent, and we have to multiply this. So we have to multiply this number by itself for another time to get this probability. So as you can see, so it's two. The chance is tiny. Yeah, it's very small. So I think probably, yeah, I don't know, like maybe I'm wrong. So there are only 4 chromosomes in watermelon. So that's why, yeah, I use this 4 chromosomes to in this example. So I need to check that. So I don't know how many chromosomes are in watermelon, but yeah. So I will check that later. I hope this is clear. So do you have any questions about this? Okay, good. All right, let's look at the tetraploid. So we talk about the tetraploid. For each chromosome, there are 4 copies, right? So then technically there are 3 possible ways for them to be paired together during the meiosis. Either this one, so this one, 2 of them will form a bivalent. So the other 2 will form another bivalent. Then these 2 will segregate independently, right? And all 4 will form together. Will be held together to form this one. How do you pronounce this? Quadrivalent. Okay, the 4 together. So those 2 are viable, but this one is not viable. So, and so either in this way or in this way, so this will produce a gamete with 2 copies of this chromosome each, right? So yeah, I hope everyone can understand this, and this will be useful for us to solve the problems in the quiz or in the final exam. Okay, so this let me see. So this is an example, but this example is probably too complicated. I just put it here for your own information, so you can after class you can read these slides and try to understand this. But this will not be on the test because it's a little bit complicated. But definitely for the triploid, is this? For the triploid problem like this, so this is a required. I will give you questions like this in the final, and I will ask you to calculate the probability based on the triploid genotype or the trisomic genotype. I think I'm gonna stop here for this because this is not that relevant to the coming Tuesday midterm. Now we still have like probably like half an hour. Let me go through let's see. Do I have it? Let me go through the practice test with you, if you don't mind. Okay. Okay. So the same. I'm gonna give you 4 questions with different points, and the total points that the maximum the possible maximum point is 75 for the midterm 2. As I mentioned, so the first one, one of the major topic to be covered in the second midterm is genetic mapping. So we're going to use the 3-point test cross as an example to. So here, this is example. So we're gonna use this to test you if you understand how we can use this tool to derive a genetic map for 3 genes, linked genes. All right. So the 3-point test cross will be a cross between a triple heterozygous individual with a homozygous recessive individual, right? And suppose this is the data you got. So this is the data you got. And from here, we need to do the major thing we need to do is like based on this data, we're gonna derive the map, the genetic map for the 3 genes. All right, how do we do this? So as we discussed before, because you see different numbers here, right, for the different genotypes. So the different number means like they are linked. So the first question is like, so suppose if these 3 genes are unlinked, so if these 3 genes are sorted independently, if they are on different chromosomes, they are unlinked. So how many progeny would you expect to see in each category, genotype category? So if these 3 genes are unlinked, then you're gonna see equal numbers in each category, right? So if we have like a total of like 1,000 progeny, then in each category, we expect to see the same numbers here. So you just divided this total number by 8. So this will be the answer for the first question. So this is a hypothesis. So if these 3 genes are not linked, this is what you expect to see. However, this is not the truth, right? Because you don't see the equal numbers in each category. So this is an indication that these 3 genes are linked. They are in LD. So what is LD? So the linkage at this equilibrium. This is a very important concept. I may ask you this question in the midterm. Okay, so next, so what is the map distance between A and D? So before you answer this question, you have to derive the map first, right? So how do we do this? So so in this data table, you see these big numbers here, the 391, 381. So the reason why they are big, these numbers are big, is because they are parental type. There's no crossover, right? So you immediately figure out the parental type is this, and you see some very small numbers here compared to the other genotype classes. So 6 and 8, so they are significantly smaller, which means they represent the double crossover categories. All right, so I have taught taught you how we can compare the double crossover type versus the parental type to figure out which gene is standing in the middle, right? So if you compare this one with this one, so only D has been changed. So the single gene that has been changed must be standing in the middle. So which means, okay, so if you want to derive the order of the 3 genes on the same chromosome, gene D is in the middle. Let's see, yes. And once you got this, you can easily, so for example, if I ask you to calculate the map distance between A and D, so you know the gene, you know the parental type of A and D, right? Little a and big D, or the big A and little d. Then you look into the other categories to figure out the non-parental type. And then you can easily calculate the recombination frequency using the number of the non-parental type divided by the total number. This gives you the genetic map between gene A and D, right? It's not that difficult, right? It's very straightforward. So as long as you understand the logic behind, so don't worry about the midterm. Yeah, you should be doing very well. All right, so this is the regular diploid organism. We can do the mapping for the neurospora as well. This is a little bit different because in the neurospora, we have two genes. We have the third gene is actually the centromere, so the same. Still, we are dealing with 3 genes, but the difference is that in neurospora, so we treat the centromere as another gene, right? So this is the same, like the 3-point test cross. All right. Okay, so if I gave you information like this, so this is a fusion. This is a temporary. So the 2 parents are coming together, the haploid neurospora, and this is the genotype. So what are the genotype for the parent neurospora strains? Of course, the genotype will be this and this, right? So if you write this, then you're going to get the credits. Oh, these are the 2 parental genotypes. Okay, now what if so what spore genotype will result if a single crossover occurs between the first one? If a single crossover occur between the N locus and the centromere, which means that there's a crossover occurring in this region. So for example, this one and this one, if there's a crossover, so what happened here? So this will be switched with this one. So but it will not change the relationship between M and N, right? Because the crossover, if occurs here and here, this crossover will not change the relationship between M and N. So the resultant progeny, the spore, resultant spores will be parental type. Okay. All right. Now, if there's a single crossover occurring between M and N, so here, let's say, then this is different because if a crossover occurs here, so this will change the relationship between M and N, right? This will give you the tetratype because if you can write the resultant genotype after the crossover, you're going to see 4 types here, 4 genotypes here. And if there's a single crossover occurring in this region, so this is totally irrelevant to the M and the centromere, right? So you do not see. So if there's a crossover occurring in this region on the left side of the M gene, then you do not see any changes in terms of the relationship between M, N, and centromere. So still gives you the parental type. All right. However, if we look at single gene, the relationship between single genes with the centromere, then that's different, right? So if a single crossover occurs between between centromere and N, then how do you specify the pattern? So for M, what is it M1 pattern or M2 pattern? So now you need to understand what M1 and M2 means, right? And so it's very easy. So I don't want to spend much time on this, but this is very important for the midterm. Okay, finally, so if we do a test cross, so this is similar to the 3-point test cross. If we got the S psi like this, so this is the number of the psi with these genotypes. Okay, how do we calculate the distance between genetic distance between the centromere and the gene N, right? It's easy. We use this formula, right? If we want to calculate the genetic distance between any single gene with the centromere, we need to figure out the number of the SI showing the M2 pattern, and so this number will be multiplied by 1/2 because only half of them represent the crossover, the non-parental type. And once you calculate this, you divide this number by the total number of the SI. This will give you the genetic map between this gene and the centromere. So very similar. So if we want to calculate the genetic distance between M and N, so we use a slightly different formula here. So we need to find out the non-parental type, because in the non-parental type, all the progenies are non-parental type, are represented crossover. So that's 100%. Okay, but the tetratype, if you look at the tetratype, only half of them represented a crossover occurring. So that's why you're going to multiply the number of SI of the tetratype by 1/2, and then you add up these two numbers, divided this number by the total number of the SI. This will give you the genetic map between these two genes. Okay. So hopefully, you can see this is important, the 3-point test cross. So spend some time on this. And also in the Neurospora, the 3-point test cross is also important. And for the second part, another major thing is to use the mutant as a technology to study the biological pathway, right? So we have learned this before recently. So if I gave you a table like this, so we have like 5 different mutant here, and each one has a single gene mutation. So this is a simple situation. So for each mutant, there's only a single mutation occurring on a single gene, and all these genes are in the same pathway like this. So, but if I give you a data like this, how do you figure out the order of the pathway? And how do you figure out for each mutant which step has been mutated? So we have learned this before, right? I showed you how we can start from this table. So we need to reorganize this table a little bit, either the column or the row, to help you can easily figure out the complete pathway for this. And all right, so this is the complementation test. So for different mutant, so first you need to figure out for each mutant which genes has something wrong, right? And then if we fuse different mutants to form some heterokaryon, so which one can grow on the minimum median, which one cannot? So this is another point with important topic we want to cover in the second midterm. All right, finally, so this is the variant of the ratios, right? So the classical ratio is 9:3 to 3:1 ratio. That represented the no interaction between these two genes. However, if you see different ratios, this is a simple example. So I just give you a simple question to try to find the relationship between these ratios and their mechanism. So in the test, maybe I can ask you to describe or give me an example to show the specific mechanism and which ratio will this mechanism will result in, so something like that. So that means like you need to spend some time to understand the mechanism, different mechanism behind the different ratios, right? And of course, finally, I'm going to give you some question like this. So I will give you some concepts like this, and you need to understand them and give me the description of each concepts. All right, so that's the review for the practice test for the second midterm. So any questions that I can help? Yeah, since we still have a little bit of time, so you can ask any questions. So maybe I remember last time a student asked me a question, but I didn't have time to talk about that. So if you prefer, so we can talk about that question now. Any questions? I can help now. Good. So in case you have some questions tomorrow, we have office hours. Lei has office hours in the morning, and in the afternoon I will hold my office hours. So if you have any questions regarding the lectures or the practice test or some aside questions in each lecture, just bring those questions to me. So we will be happy to help with those questions. All right. So I'm gonna stay here for a little bit while. So in case you have questions, if not, you feel free. You feel free to go. All right. Thank you, everyone.
Hi, Professor.
Yep.
For number 2 on the practice midterm, can you go over question 3 for that?
Yes, sure. Let me just... Number 2, right? Question 3.
Yes.
Okay. Yes. Okay. Sure. Okay. So now the number 2 actually is focused on the relationship between 2 genes, right? But number 3 is the focus of number 3 is to talk about the relationship between each gene, let's say N, with the centromere. Okay. Now then, if we talk about the relationship between the gene, single gene, and the centromere, then we're gonna use either the M1 pattern or M2 pattern. Okay. So in the first situation, if there's a crossover between N and the centromere, which means here. So if there's a crossover between here and here, this will change the relationship between centromere and the N, right? So if there was no crossover, then this is an M1 pattern because the first meiosis step will so you will see the segregation between M plus and N in the first meiosis step, right? If there were no crossover. However, if there's a crossover, if there is a crossover between N and the centromere, then you're gonna see you're gonna only see the segregation between M plus and N in the second step of the meiosis. So that's why this is an M2 pattern, right? For N, and the same for M, because if you have a crossover here, so you will change the pattern between the centromere and N. You will simultaneously change the pattern between the centromere and the M. So for both of them, they are M2 pattern. However, if there's a crossover occurring between M and N, so here, then this crossover only changes the relationship between M and the centromere, but not N and the centromere. So that's why if you look at the M, so it is M2 pattern, but when we talk about the relationship between N and the centromere, still this is M1 pattern.
I'm
And
a
so on.
little confused still. So is that gray dot the centromere, or is it the space between the 2 strains?
Yeah. Okay. So yeah. Okay. Maybe it's better for me to draw something on the slides. It's easier to. So give me a second. Let me remember this diagram. Okay. Let me stop share this first, and probably it's better to use this. And let me use a new slides layout, like, okay. Okay, so this is easier for me to explain. Okay, so this is the neurospore, right? We have centromere. This is the first parent, centromere, and this is M plus here, M and M. So now we have another parent. This is N and this is N, right? This is M plus, M plus. Okay. All right. So let's look at here. So if there's a crossover occurring between N and the centromere, so then after the crossing over, you're gonna see what? You're gonna see this. You're gonna see, okay, so this is still M plus, this is M, right? But here you're gonna see this because they're changed, right? So this will become N and this will become M plus. All right, now since there's an exchange between these two parents, you're gonna see this. So for this one, it's unchanged, but this one is M plus and M, right? Okay, now if this goes through the meiosis, meiosis 1 is what? Is this two is gonna separate, right? All right, this will become 2 cells, right? Because these 2 homologous chromosomes are gonna segregate. Did you see? So in each of the daughter cell, so this is one daughter cell and this is another daughter cell. Did you see the segregation of M plus and N? No. So they are still together in the same cell, right? So that means like this is not a M1 pattern. So this 2 only can be segregated if this cell go to the second stage of the meiosis. If this one separate with this one, then they can segregate. That's why this situation, the gene A is a M2 pattern, right? Is that clear? No? I can redo this. So probably, yeah, this is a little bit messy. Okay, let's do this. Okay. So first, we need to let me use the slides. I think it's better to use the lecture slides to discuss this. Let's see. I believe it's lecture 7 and 8. Pattern 1. Oh, okay. No, 9 and 10, I think. It's 9 and 10. Okay, yes. Okay. Nice. Okay, so this is better. I think, yeah, it's better. So it's important for us to understand the difference between M1 pattern and M2 pattern. Okay, all right. So if there's no crossover, so this is one parent, right? This is another parent. So we want to see at which stage, at which step in the meiosis, these 2 alleles are gonna, the big A and the small a, are gonna segregate. So definitely in this initial stage, you're gonna see the big A and the small a both in the same cell. So they didn't segregate, right? So this is this situation. Now let's look at the first meiotic division. Because there's no crossover, so this will end up into 2 different daughter cells, right? So we did see the segregation between big A and the small a because big A or big A goes to one cell, and or small a goes to another cell. So this means like the two alleles segregate at the M1 pattern, at M1 stage, right? The first meiotic stage, right? Okay, now let's look at this. So if there's a crossover between these two parents, then what happens? This is the first division. Did you see the segregation? No, because of the crossover. So the big A and the small a both go together to this cell and this cell. So you do not see the clear segregation between these two alleles at M1 stage, right? Only if this cell enters into the second stage of the meiosis. So here, then you see the big A, small a segregate. They goes into different cells. Therefore, this is the M2 pattern. Is that clear? And this is the relationship between this gene and the centromere. Yeah, I hope I answered your question.
So for the N and centromere, they're both M2 because once they segregate, like
Right.
the M plus and like the M plus and N they're like all switched up. They're not together.
Yes, yes, exactly.
Okay. And then for M and left end of chromosome, I was a little confused on what you defined as the left end of the chromosome.
Which one? Let me see.
For C for 2 and like the third one
This?
for yeah.
Oh, right. So here when we talk about okay. So let me show you. So this is interesting. So only the crossover occurring here or here can mess up the initial relationship between M and N with the centromere, right? If there's a crossover occurring here, which means, yeah, this region will not be affected, only this region. So think about here. So only the region on the left will be exchanged between these two, So it will not affect. It will not affect the relationship between M and N with the centromere. So let me draw this picture to you. Presentation. Okay. So okay, this is a good question. Okay, let's say this is the first parent. This is M plus M. So let's see, this chromosome is very long. Okay, very long.
I'm not sure if I can see your screen or if you're drawing anything.
Huh?
Are you drawing something? Because I can't see it.
Oh, you cannot see it? Can you see it? I'm sorry. Can you see my slides?
No.
Oh, sorry. Okay, let me redo this.
It still shows the.
Okay.
How about now? How about now? Can you see it?
Yes.
Okay, sorry. Yeah, okay. So this is the question, right? All right. So if there's a crossover occurring on the left-hand side of M, let's say here, if crossover between these two chromatids in this region, so only this region will be switched with this. So this change will not affect the relationship between M and N with the centromere, right? Only this region, 1 and 2, will be swapped. So this crossover will not affect any patterns between M or N to the centromere. Does that make sense?
So it's just completely unrelated to M and N?
That is true. Yeah, that is true. That's the point for this question. Yes.
Would it be possible for there to be a crossover between N and the left end of the chromosome? Or does it have to by something to cross?
Oh, yeah, that's possible. Because here we only talk about this gene, right? There's a lot of genes on this region as well. So it is very likely that a crossover occurs in this region, which are totally irrelevant with these two genes with the centromere.
Okay.
Yeah,
And
because
then-
here we only focus on these two genes, right? But if the crossover occurring in this region, it will change other genes, but it will not change these two genes, right?
Mhm.
Yeah.
Okay.
Professor,
then
what's
for-
the name of this kind of problem?
Mhm.
Like, what are these kinds of problems called specifically? Like with the near
What this
square
kind
and-
of problem called? You mean this?
Mhm.
This is so I would like to say this is also so this is a special case for the 3-point test cross. When we use the diploid organism to do the mapping, genetic mapping, so we use like big A, big B, and big C, right? Small a, small b, and small c. So this is the 3-point test cross in the diploid organism. Now, this is a special case. Still, we focus on 3 genes, but the third gene is centromere, not the gene, right? Real gene, but still, yeah, you can treat them the same. And this is in the neurospora. So we use a haploid model organism to handle the genetic mapping. Same, the genetic mapping problem. Did I answer your question?
Oh yeah, you answered it beautifully.
Okay, perfect. Thank you.
Professor, I had a quick question. I'm not sure if you mentioned this before, but are you still going to hold the genetics clinic, or is it just going to be office hours?
Oh yeah, it's the same. So the genetic clinic will be like tomorrow. I will have a genetic clinic, but that's actually technically that's an extension of my office hours. Usually I'm gonna stay in my office hours to answer students' question, but in the past I rarely see students coming, so then there's no point for me to stay there for a whole 2 hours. So, but if there are a lot of students coming for questions, so if 1 hour is not enough, so I'm gonna use 2 hours, like another extra hours from 4 o'clock to 5 o'clock tomorrow to answer your question.
Well, rest assured, Professor, I'll be there, so it won't
Oh,
be
okay,
totally
good.
empty.
See you
Yeah,
tomorrow.
that makes sense. Thank you, Professor.
Sure. Thank you. So any other questions?
I guess I'll ask you them tomorrow during your office hours in genetic clinic. Thank you, though.
Oh, sure.
Have a great
Okay.
day.
You too. Bye-bye. See you tomorrow. Bye-bye.
See you. Bye.
Bye, everyone. Bye.